Yeah, thank you very much. So what I would like to do this afternoon is to give you an overview of what has been done so far with microarrays in the area of ventilator-induced lung injury. And before I go into those very few studies that have been done, I would just like briefly to give you an overview of the microarray technique. Next slide. Sorry, I'm pressing here and nothing happens. No, but we have another arrangement. So, okay, now. It's not here. Oh, I'm always looking here. It's only here. Okay, good. So I stopped looking here. Okay, so this is just an overview of different screening methods that are recently being used, and probably you've heard of all of them. So if you have the classical dogma of molecular biology going from DNA to RNA to protein synthesis, now we have screening methods at all levels. So at the DNA level, of course, we have the genotyping and the SNP analysis. At the RNA level, and that's what microarrays are used for and what I will be talking today, we have the gene expression arrays, which is another word for microarrays. And then, of course, at the protein level, we have the proteomics. We have protein profiling, as it's called here, and also biomarker detection. So what I will be talking today is this area of gene expression study. So what microarrays really are for is to figure out which genes are turned on under specific situations. And of course, the great advantage of microarrays is that you really can have basically all the genes, all the 25,000 genes that we have, and look at their expression. So that's an enormous amount of data you can generate. And of course, there are three basic reasons why you would like to do that. The first one is you would like to find new candidate genes. So you would just like to see whether there is another gene that you overlooked with the traditional methods that could add, for instance, to ventilator-induced lung injury. Another thing is now it becomes actually possible for almost the first time to study patterns of changes. So really to see that there is a whole array of genes that is changed at the same time. And of course, this is more obvious during developmental biology or also maybe during cancer, which are long-term processes. It's more difficult to see those patterns of changes really in events that happen very, very quickly, like sepsis or ventilator-induced lung injury. But clearly also there are such patterns, but they are more difficult to understand right now. And of course, all this needs then to the creation of new hypotheses. And in the area, here we go. And in the area of ventilator-induced lung injury, I think there is one specific point why microarrays could be particularly valuable, and that is shown on this slide. So what is shown here is that we can have different stimuli. So this is overdistension and then a mechanosensor that we're not completely sure of what it is right now. On this side, the overventilation mechanosensor. Here we have the microorganisms, and then that stimulates the toll-like receptors. And basically what we've learned in the last years is that both stimuli really can lead to the activation of a common pathway of inflammation. So here we have transcription factors like NF-kappa B I talked about yesterday, also AP1. And those that have been particularly shown here is the J&K, for instance, as an example, where it was shown to be involved in ventilator-induced lung injury. Then, of course, these transcription factors lead to the production of products, so of cytokines, adhesion molecules, chemokines, and examples of that have been shown to be important in animal models of ventilator-induced lung injury are the MIP2, which is a chemokine similar to the human interleukin-8, selectins, so adhesion molecules, and human necrosis factor. And then this leads then finally to the recruitment and activation of neutrophils. So in all those animal models of ventilator-induced lung injury, or most of them at least, finally you have invasion of neutrophils into the lung tissue. And then the concept is that those neutrophils, if this is of course stimulated by bacteria, the idea would be that they cause or help to kill the bacteria. However, we also know if the neutrophils become overactivated, this may also lead to acute lung injury because the weapons that the neutrophils have are very unspecific. There are so many different bacteria So it's impossible for the neutrophils to have a specific tool for every bacterium. So it uses reactive oxidant species, proteases, and so forth. And these are very unspecific weapons that also damage the adjacent lung tissue. Having said that, we know from a lot of studies that even though in animal models we can interfere here successfully, the clinical trials that try to block inflammation at various steps, be it in sepsis or acute lung injury, actually, none of them was really successful. So one reason, there's a couple of reasons, but one reason could be that probably it's not prudent to block the innate immune system, because this, of course, can also cause immune tolerance and make the patients more susceptible to infections, for instance. However, what could be possible, and I think that's where microarrays can really help us a lot, is we know, like Art pointed yesterday, we really know when we start the ventilation. And I think for me, it's very clear that mechanical ventilation or ventilation of lungs that are already pre-injured is such a delicate task that it's almost impossible not to do additional harm to the lungs by that. So this may be bigger or smaller, that's one thing, but definitely it's a huge problem to ventilate an already injured lung. But the advantage is we already know when this starts. So what might be feasible is that we find ways, signaling ways or genes that are specifically turned on by mechanical ventilation. And if we were able then to block those genes that also contribute to neutrophil recruitment, for instance, in acute lung injury, then we might have a way to specifically block the side effects of mechanical ventilation without blocking the innate immune system. And I think this is an area where really microarrays can help a lot, because they can single out those genes. So if you make a comparison between activation of the innate immune system by bacteria or by acid installation on the one hand and compare it to ventilation-induced lung injury, you can pick up genes that may be specific. So now let's just a very, very brief overview how microarrays are done. The reality is somewhat more complicated, but this is a basic picture of what you are doing. So what you do is you isolate the RNA from your samples, for instance, from the lung tissue, from cell cultures, actually every source you would like to that has RNA. And then you hybridize the probes that the RNA that you have that you received here, you hybridize it with a chip. And this chip has spotted a huge number of DNA on it for a huge number of genes, and this can go up really to 25,000, 30,000 genes being spotted on one single chip. And then you hybridize. And then after you hybridize, you can see which of your RNA probe, which of the RNAs you hybridize to what extent with the DNA, the cDNA that was present on the chip. So actually, it's nothing really complicated. It's nothing new. We've known these hybridization techniques for a long time. What is new is now that we are able to do this on such an enormous scale. And of course, there are a number of technical problems occurring there, and I just want to briefly mention those. Of course, it's not, because the first step, once you've done all the biology and the hybridization, which has its own biological problems, of course, after that, you have to take a digital image of the microarray. And of course, everybody who's doing photography and who's working with Photoshop is knowing what sorts of problems you can have with pictures. So you can have unequal lighting, so one part of the picture may be a little bit blacker or darker, and all these sort of things. So the whole problem of digital imaging is a very important issue here. The second thing is you have to have homogeneous hybridization. So what you really assume is that every gene has the same chance, or that every RNA has the same chance as another gene to hybridize. However, this is very difficult really to verify, because the probes that you construct, I mean, you have 30,000 probes, so you cannot really show that all of them have the same quality. So there is an inherent problem that the hybridization of one gene may be better than the other. A further problem is the normalization of arrays. So let's say you do 2 or 4 arrays, one control array, one of a ventilated lung, and so forth. Now you want, of course, compare which genes are being turned on. So you have to make sure that the signals you get are comparable. So you have to normalize those arrays. And that's, again, not a trivial task. How do you normalize? How do you make sure that that really the signals you get are the same. Of course, there are housekeeping genes, for instance, of which you assume that they are constant, and then you could normalize to those, and so on and so on. There are a couple of possibilities. I don't want to go into that. Just this is really a big task to do this properly again. And then finally, another big issue is the multiple testing. You all know is the more hypotheses you put up, the more likely to find a true one in statistical terms, even though it's not true in reality. Right? So you know we always have the 0.05 level in our significance. So that means that we allow one false positive result out of 20. So now we have 30,000 genes. So clearly, we'll have a lot of genes showing up just by chance, even though they are not changed in reality. And there are pretty clever ways now to deal with this problem, but in essence, of course, what you always have to do in the end is you have to verify the data from the microarray with other means. So with PCR, for instance, best would be to generate new samples, and so on. So these are certainly problems, but as time is going on, the solutions for these problems are becoming better and better. So now coming to the area of ventilator-induced lung injury. And as far as I know, there are only a few studies being performed in this area, and I've summarized them here, and I go a little bit through all of them. So of course we have different levels on which such experiments could be performed. So we have cells that we can stretch. We have explants or lung slices, as is small tissue slices from lungs. We have isolated perfused lungs, and of course we have the in vivo model. And I just would like to show you one or two data from each of those studies, and then discuss the advantages and disadvantages of those techniques with you. So this is a study that was done from Claudia dos Santos, also together with Ming-Yao Yu in Toronto and Art Slutsky. And Ralph Hubmeier was also an author on that paper. And actually what was done in this study, Claudia tried to stretch A549 cells, which are a cancer cell which in some respects mimics pulmonary epithelial cells. So I think the phenotype of this cell is not really clear. And there have been some studies that showed, if you look for single genes, that if you stretch those cells, you can induce interleukin-8, for instance, stimulation. However, in this area, actually, even though you can see that here are 3 genes, even though TNF was clearly able to induce a number of genes, actually stretch had no effect on any of those genes. Although, as you can see here, stretch was clearly effective. You see the morphology of the cells was different after they were stretched. And also, what you see here, there was an activation of a protein, ZARC. Doesn't really matter what it is, but it's known to become activated during stretch. And this was happening too. So these cells clearly responded to stretch, but we couldn't really see any increase in gene expression. And I think this may be a problem at the I549 cells, which may differ from lab to lab, but also the phenotype of which is pretty strange. For instance, they respond very poorly to endotoxin, for instance. They have no receptors for endotoxin, although normal epithelial cells have. However, of course, the reasons why people are using those cells is that really we don't have a good alternative at that time. And I think that's a major point. Of course, we would like to stretch cells, because stretching cells would have a lot of advantages. We're very controlled with conditions. The manipulation we have at hands are much more potent. However, at this time, it's very difficult to do, because we really don't have good cell lines in the lung at this time. This is really an effect that hinders a lot of research in this area. The cell lines are not really good. It's not easy to get primary cells, and the primary cells in culture and the type 2 cells, they differentiate pretty quickly, and it's not easy to keep them stable, and so on and so on. And there recently has been a study that shows even if you take type 2 cells into culture, the phenotype is completely different. If you make a gene array, the phenotype is completely different than the phenotype of the original type 2 cells. So there are lots of problems here. And one further problem that I would like to mention is that even though if you stretch cells in culture, the real amount of stretch that occurs to these cells in vivo is completely unknown. We have really no good, and Gary gave a beautiful presentation yesterday of the different models, what happens when the lung is breathing. And there are all kinds of ideas. So it could really be that the alveoli distend, but it could as well be that as Gary is saying, that the alveolar ducts are increasing. So actually, we really don't know which cells change in size, and we have no idea to what extent. they do that. So at this time also it requires a lot of assumptions to take cells into culture and to stretch them. And this is just to give you an example of studies that we did to address this issue of different cell types. So these are pictures from an isolated perfused mouse lung, and this is a picture of the alveoli that we see with 3, 10, and 25 centimeter water column, and you can clearly see how in our model alveoli are increasing under these conditions. And what we have seen is now that NF-kappa B, for instance, yesterday I introduced you to this transcription factor, becomes activated in alveolar macrophages and also in type 2 cells. Here you can see the activation of the NF-kappa B. And just coming back to the point of difficulty is we have no idea how these cells get stretched. I think it's an extremely interesting question. How does the alveolar macrophage become activated under these conditions? Because the alveolar macrophage basically sits in the alveolar space. But so how does it know that the alveolus get stretched if it's getting stretched at all? So we really don't have an answer to that. And it may very well be that the alveolar macrophage is not really getting stretched at all, that it's indirect signaling. So for instance, if we take alveolar macrophages into culture and stretch them, we really don't know whether we are looking at the real thing. So an alternative to that would be, one step further would be to try to stretch intact lung tissue. And we haven't done that yet, but this is something we are planning. So there are no studies, no microarray studies yet. So Martin Post and Ming-Yao Liu in Toronto, they have been stretching lung explants for a long time and have a lot of experience with that. But they haven't done really, as far as I know, at least a microarray on that. We have a slightly different slice model. These are slices from adult lungs. And you see here those slices. This is a petri dish to give you an idea of the size. Here you can see an airway in the middle. And the advantage of this model is that we really can prepare those slices. We call them precision-cut lung slices because they're about almost precisely 220 micrometers thin or thick, as you like. And one advantage is that you can get those slices from almost every species. So we have slices from rats and guinea pigs, recently also from monkeys and also from humans. And of course, this would provide probably the only way to stretch intact human tissue. And that's something we are going for in the future. And just to show you that these slices are alive, I would like to show you 2 movies. So that's an airway of such a slice, and this is passively sensitized. We put allergen on it. And then you can see that in a time frame of 10 minutes, you can see how the airway contracts. So these airways are clearly alive, and I think it would be very interesting to stretch them and then to analyze by microarray and by other tools what's going on here. So the advantage of the explants would be that the microanatomy is intact, we can use human tissue, and we can also define the stretch pretty well. Of course, there are also disadvantages, one being that, of course, there is no nervous system and no interaction with other parts of the body, so we clearly also lack some of the complete picture here in this model. The next model you could use would be isolated perfused lungs, and these are isolated perfused mouse lungs. You can see this is ventilated with 25 centimeter water column, this with 10 centimeter water column. And this is a model that we use a lot to see which genes are being turned on under this condition. And here you see now a movie of the alveoli on the surface of those lungs. And you can see that there is certainly stretch occurring to a lot of those alveoli as they get bigger and bigger. And of course, we are very interested which genes are being turned on under these conditions. And we have recently done a trial together with the group of Augustine Choi in Pittsburgh and looked for a number of genes in this model. And what we did was we compared the control lungs to the toxin-treated lungs and to lungs that were ventilated or overventilated, so with 25 centimeter water column. And I think a major point from here is what you see is, of course, these are all different lungs, and of course the intensity here tells you whether the genes are being up or down regulated. And I think what is very evident is that endotoxin and overventilation, there's a lot of genes that are regulated in the same direction. And when we calculate a regression coefficient, we see that we get a coefficient of about 0.4, and the probability that there is a correlation is 0.0001. So many genes are turned on similarly by endotoxin and by overventilation. But of course, we are interested in those genes that are specifically turned on. And one of them, and actually I decided not to give you big lists of genes, because probably many of you have seen microarray talks before, and then usually you get a list of genes written smaller and smaller because you get so much information, but in the end, really, if you're in the auditorium, it doesn't really help you, because you cannot write them down. So I think this should be, if you like to see all those genes, you really have to go to the papers. So I just picked up one, which I think is particularly interesting. This is amphiregulin. And I come to what it does in a moment. So what you see here, and this now is the PCR. So this is the gene expression, and this is a protein in the western blot of those lungs. And you can see there is some activation by endotoxin, but only very little. However, in response to overventilation, there is a huge increase. And also this has been confirmed by immune histochemistry, and it appears to be primarily epithelial cells that become activated and display amphiregulin. And this is a candidate gene. This is a gene that would help us to identify or to have a new hypothesis. And actually, what's this here? Sorry. I'm a little bit, there should be, 1 second. Oh, somehow there's a gene missing, there are slides missing. 1 second, that's strange. Let me just see what's going on this one. Because one second. Because we had the problem with two versions, and it seems like the old version I had before on that computer is still there. Actually, that's what's shown. Let me just make sure that we have the right one. Yeah, on my computer here has the correct one. Can I continue with this one? Okay. This is the right one here, I would like to show. Okay, so let's continue with this one. Okay. So now what you see, that's what I was going to show you. So these are genes being increased during overventilation, during LPS, and I just took out the area where the increase in expression is not as strong, because obviously if you include these, you will have a much, much stronger correlation, because if genes don't change at all, I mean, it's easy to get a good correlation. So I took them out, but you see, even then, the correlation is pretty good. So many genes that are activated by endotoxin are also activated by stretching those lungs. And then we had the amphiregulin I taught you before. And actually, amphiregulin is very interesting because and that was one reason why I showed you this pathway yesterday, because amphiregulin is another molecule that, like the HBEGF, is expressed on the outer side of the cell and then is cleaved by metalloproteases, as I discussed with you yesterday. So one hypothesis, obviously, that would be generated from that is that maybe during ventilator-induced lung injury, if you stretch cells, what happens is that not HBEGF is really being released, but it's amphiregulin instead, but then you would still enter a very similar pathway, and also amphiregulin is an activator of the EGF receptor. So I think that is an excellent example how hypotheses can be generated from microarrays, because otherwise we never would have thought of amphiregulin. And of course, now we have to figure out in further studies how specific amphiregulin really is for ventilator-induced lung injury and what role it plays. So, summarizing what you can do with microarrays in the intact lung is you have the intact organ, which clearly is an advantage. So the microanatomy is maintained, and you know that you are stretching the alveolar in a realistic and meaningful way. You have no blood cells, and of course you may think that is a disadvantage, but this of course also is a huge advantage. Because if you do microarray studies in vivo, what you're going to do is you also measure the genes of the neutrophils that moved into the lungs. So if you see an increased gene expression in vivo, some of the genes may only be increased because you have a new cell type in the lung. So in some way, if you're not careful, the microarrays could be a very expensive way of measuring neutrophil infiltration. Okay, so that's why I think the isolated organ is a very important additional information to have. So then you can see which of the genes that go on because of stretch and which are neutrophil related. However, of course, we have no nervous system again, and we don't have the opportunity realistically to use human tissue. Now coming to the in vivo work, and there are two studies. This one is from the group of Skip Garcia, just published in the September issue of the American Journal. Sorry, of the American Journal of Physiology. And this shows another way of looking at the data. And what they figured out was this gene, the MYC6 gene, which also is called gene 33, which really had deserved very few attention so far. But it was upregulated a lot by over-distending those lungs. So once they made this observation, they asked, can we have a relation of these genes to genes that we already know? So this is called a relevance network. And there are computer programs to do that for you now. So you have a gene, and then you ask which of the genes that you measured are related to the one that you have here. And then you get a picture like that. And you see that in the works, you see, for instance, that this is related to the plower gene. So these are all the gene symbols now. So this is a urocinin plus minogen activator receptor 1 gene. So this would be activating fibrinolysis. And actually, it's an interesting observation that in many of those gene areas of overventilation, you see an activation or changes in the clotting system and fibrinolytic system. So that clearly plays an important role, which, of course, is a very interesting observation. As you know, because acute lung injury also has been termed hyaline membrane disease, which clearly has to do with clotting. So the gene areas point to that. And you see a number of genes here. I don't want to really go through all of them. You see there is a path from MYC6 through a transcription factor that we hadn't on our list before, ATF3 to interleukin 6, for instance. They also see in their study an upregulation of amphiregulin and so forth. So from this, of course, you can again create a whole bunch of new hypotheses and then study how these different genes interact with each other. And what I did here is I just compared some of the genes that they found increased in the microarray in the intact mice or rats, which are shown on the y-axis, with those which we found in our isolated perfused mouse lung model. And you really see that the correlation is quite good for most of the genes. Actually, and those were all the genes that were significant, right? That were significant in their array and in our array. Of course, there are much more genes, but there are special ways we use a SEM program to see which genes are significant. They use the same. So these are only genes we are really sure of they go up. And you see the correlation is pretty good. And there are also some genes that go up in vivo, but that do not go up in the perfused lung. And one reason why this might be so is that these genes are related to neutrophil infiltration. So the advantage, of course, of the in vivo models is that you have a complex model. It's a full complexity. However, problems are, again, no human tissue is possible, can be studied. And you may also study genes from infiltrated cells. And the last study that I would like to discuss with you is a study that I did together with Art Slutsky when I did a sabbatical in his lab. And most of the work really was done by Yumiko Imai, who is now together with Josef Penninger in Vienna. And what we did there was we studied a 2-hit model and then made gene area on that. So the first question is, which genes are being activated during stress induced by acid inspiration? And then the second question was, if you now ventilate those lungs, what does happen? Is there an additional genes? Are there additional genes being turned on? And that is shown here. So we had a normal group, there was a sham group, and then there was a group that received acid and was ventilated with a B=1 story that Marco Ranieri introduced to you yesterday. Then there was one group that was activated with a strategy that would over-distend the lung, defined by the B/1. And then there was another group which is sort of, which unfortunately is missing here, B=1. So we also had de-recruitment. So we had 3 different conditions. And this was a strategy which we assumed to be a protective ventilation strategy. And then we had 2 strategies, one over-distension and one de-recruitment, to see which genes are being turned on. And these are, I think, about 3,000 genes in each row that we looked at and just put them into a mathematical analysis which is called cluster analysis. And this cluster analysis really gives us a very nice result because you see the clustering of the different genes which are activated under those conditions. You see it's clustering the sham and the normal animals together, and it's clustering the acid-treated animals together. And again, the correlation between the 2 strategies that are injurious ventilation strategies are again closer. So this, of course, is a very nice result, which is also displayed if you look at the elastance. So the elastance is a marker for the stiffness of the lung, and the higher the elastance, the greater the stiffness of the lung is, you know. And you see, with both B/1, so with over-distension and de-recruitment, there was an increase in elastance. There also was an increase in neutrophil infiltration in the lungs and so forth. So there was acute lung injury. And the B=1, the protective ventilation strategy, those lungs look pretty good, actually. So then we looked at different genes under these conditions, and we started with this gene just to give you some numbers, so you see how you go down with numbers in a real assay, in a real array. So we started with 8,799 targets on chips. And I call this targets because on many of those chips, genes are present 2, 3, 4, or even 5 times. So if you have 8,800 targets, it does not mean you're looking at the same number of genes. And sometimes it's a problem to figure out really where all those common genes are. So of those of those targets, 3,200 roughly were present in the lung tissue. Does not mean that they were changed at all, just the probes hybridized on the array. Then we used an analysis which is called SAM, Statistical Analysis of Microarray, which is a pretty ingenious way to deal with the problem of multiple testing. And then 467 targets were left. Looking at those 467 targets, we identified that those really represented only 383 genes that were regulated in one way or the other, and 164 of them downregulated and 211 of them upregulated. And then one way to look at those data was to make a plot like this. And the idea of this plot is that we would like to know which genes are activated by overdistension, and they are shown along this axis, and which genes are activated by de-recruitment, showing on this axis. So remember that if it's one, I mean, everything was normalized to the animals that had protective ventilation. So all these animals had acid. Okay? So a number of these genes were already activated a lot by acid, but then were further activated by either overdistension or de-recruitment, and that is shown here. And of course, those genes that now we are really interested in are those genes which are really in this area, which are strongly upregulated by either de-recruitment or overdistension, or by both. And I will give you only one example and then show you how we pursued this example, and this has to do with chemokines. So we realized that we have a lot of genes associated with inflammation were activated, and many of them also chemokines. So you see here the whole tree of chemokines that we have, and this system, as you will know, is very redundant. We have a number of receptors, but we have even much more ligands to those receptors. So it's a very complicated system. And actually, I think to understand such a system, you have to have screening methods like the gene array to get at least a grasp of this complexity. So what you see here is everything which is circled in orange here, MCP1, MIP1 alpha, MIP3 alpha, interleukin 8 or MIP2, all of them were increased by mechanical ventilation. And one gene I would like to focus on now, this is the IP10 gene, which is shown here, because this was activated a lot by acid, but even more on that by de-recruitment or overdistension. And that is shown here. So this is the gene expression now. I don't show you the gene array data, but this is the gene expression now, and it's a protein measured in those lungs with ELISA. And you can see the data are pretty nice going together. So this is the sham lung, the normal lung, the sham lung. So there's no IP10 in those lung at basal levels. If they are treated with acid, there is an increase. And if you ventilate those lungs with a de-recruitment or an over-distending strategy, there is an increase under both conditions. So IP10 seems to be a novel gene involved in acute lung injury. And the same goes here for the protein. And then we did studies with IP10 knockout mice. We also did studies with antibodies against IP10. They gave exactly the same result. I only show you data with the IP10 knockout mice. And you can see that and this is now again elastance. And this is a change of elastance compared to the acid-treated animals ventilated with a protective strategy. And you can see there was a small decrease of elastance in the knockout mouse being treated with a protective strategy, which tells us that even in the acid model, IP10 plays some role. But what you can see is that the role in the ventilator-induced lung injury of IP10 is much stronger. So in those knockout mice, the the IP10 knockout mouse, either during the over-distelling or the recruitment ventilation strategy, almost went back to the effect of this of 100% of the animals that were protectively ventilated. And of course, from that, if you further ask what is the role of IP10, and I will give you that information in a moment, and of course, is this relevant to humans? And we were so lucky as to have still samples left from the study from Marco Ranieri and Art Stratsky. I think it was in JAMA 1999. And we still had some samples left from that study, and we used those samples to measure IP10. And these were the patients ventilated with 11 milliliters per kilogram tidal volume, and these were the animals, or the humans, pardon, the human ALDS patients, the human ALDS patients ventilated with 7.6 milliliters per kilogram tidal volume. And you can clearly see that during these different ventilation strategies, the IP10 was increasing with the high tidal volume ventilation strategy, whereas it stayed constant in those patients with a low tidal volume strategy. So even though this does not tell us that IP10 really plays a causal role in human acute lung injury and in human ventilator-associated lung injury, this is strong evidence that it might play a role. So just to summarize what we think, how IP10 is working. So we think that many stimuli, like acid installation, sepsis, overventilation, they stimulate alveolar macrophages, epithelial cells, but probably also other cells to produce IP10. And then data that I didn't show you indicates that IP10 has at least two actions, because IP10 is a chemokine. However, IP10 has no direct actions on neutrophils. And that was puzzling at first. So we had to explain why lungs, or how IP10 could increase neutrophil infiltration in those lungs, what we observed, if it does have no direct effect on neutrophils. So neutrophils don't have the CXCR3 receptor for IP10, for instance. So what we figured out now recently is that IP10 has two actions. The first one is primes endothelial cells for adhesion for PMN induced by interleukin-8. So you need much less interleukin-8 in the presence of IP10 for neutrophils to adhere to those cells. And the second thing is, and this was done in human lung explants, if you stimulate human lung explants with IP10, you see that they also release interleukin-8. So both of these actions then would lead to an increased sequestration of neutrophils in the lung, and then could explain how acute lung injury is forming under those conditions. So just to summarize, going through the three different topics that I had in the beginning, why would you like to use microarray analysis in ventilator-induced lung injury? You would like to find new candidate genes, and I described three of them, amphiregulin, MYC6, and IP10. You could study patterns rather than single genes, what we have observed for chemokines, as I showed you, and also for the clotting system. I didn't show you data on that, I only briefly mentioned that. And of course, you can create new hypotheses, and this is something I haven't talked about now, but I can tell you we'll hear more about this in the future. We have good evidence that also mechanical ventilation interacts with the nervous system and causes the release of neuropeptides, and also this plays an important role in ventilator-induced lung injury. So finally, I would like to thank all the co-workers that contributed to this work, because from our group in Boston and in Lübeck, then from Toronto and Vienna, Art Stratsky and Yumiko Ima being particularly important in those studies, and then in Pittsburgh, Augustine Choi and Tamas Dolinai. Thank you very much.